Welcome viewers. Today we are going to discuss about the practical where we are going to transform E.Coli XL1 blue cells with plasmid DNA called PUC19. So transformation is supposed to be one of the important methods for introduction of foreign DNA inside the bacterial cells. This is supposed to be the method of choice for all recombinant DNA technologists to carry on the cloning of various genes inside bacterial hosts. Actually, bacteria do have three different mechanisms of gene transfer inside the bacterial cells. Number one, transformation, wherein we will see that the naked DNA, which is either being secreted by a dying cell, gets transferred onto a recipient bacteria. The fate of that exogenote is either it exists as an independent entity or it gets integrated inside the bacterial host. The second method of gene transfer is by means of conjugation. Therein we will see that there exists a donor cell which contains a plasmid, and there also exists a recipient cell which doesn't contain the plasmid. And the donor bacteria transfers the plasmid from the donor cell to the recipient cell. At the same time, the plasmid undergoes replication. So, at the end of conjugation, we will see that both the cells become equivalent to donor cells. And the third method of gene transfer in bacteria is transduction. Herein, we will see that bacteriophages that infect the bacterial cells, they destroy or rupture the bacterial genome, and while they come out after lysis of their host cells, sometimes instead of packaging their own genomes, they package the bacterial DNA into their phage heads. And during second round of infection, this bacteriophage is transferring bacterial DNA to a new bacterial host cell. So these are the three broad methods of gene transfer mechanisms that are available in bacteria. They are very much important from the viewpoint of generating diversity in the genomes of bacteria. As we will see that sometimes the external DNA that gets inside the bacterial cell may code for certain antibiotic resistance genes which helps the bacteria to survive under the antibiotic stress. Otherwise, the normal bacteria was antibiotic sensitive, but it could survive in presence of antibiotic as because it has acquired the plasmid via the method of transformation or conjugation or transduction. The bacteria gets the selective advantage of survival if they are stressed or they are allowed or forced to grow in presence of heavy metals. Then the plasmid may code for certain enzymes that help the bacteria to survive under this adverse situations. During bacterial transformation, there are certain bacteria which are naturally capable of uptaking foreign DNA. So, they are called naturally transformable bacterial strains. On the other hand, there are certain bacteria that are normally incapable of uptaking a foreign DNA. So, under certain conditions, we will see that they become capable of uptaking the DNA. That condition is called as making the cell a competent cell. Frederick Griffith, who actually discovered the transforming principle in the year 1928, he worked on Streptococcus pneumoniae. At that time, actually, he just could establish the existence of transforming principle, but unfortunately, he couldn't unravel that what is the nature of the transforming principle. So this truth was unraveled by the tremendous work of three eminent scientists, Oswald T. Avery, Colin MacLeod, and Macklin McCarty. They actually demonstrated that the transforming principle was nothing but DNA. They ruled out the probability of RNA or protein as the transforming principle. So this is supposed to be the most important milestones in the elucidation of molecular nature of the genes. By means of natural transformation, actually the mapping of unknown bacterial genomes was made possible. The known host is Bacillus subtilis, which can be transformed naturally. By means of artificial transformation only, recombinant DNA technologists have been successful in introducing foreign genes inside bacterial cells and overexpressing those genes inside the bacterial hosts. First of all, the DNA which is lying outside, it remains as a double-stranded DNA and it binds to the cell wall. There contains certain DNA binding proteins onto the cell wall which binds the DNA and allows the entry of the DNA from the cell wall through the cytoplasmic membrane. In the cytoplasmic membrane, there actually exists certain DNA degrading enzymes that convert this double-stranded DNA into a single-stranded DNA. And the single-stranded DNA then gets inside the recipient bacterial cell and sometimes it gets integrated inside the bacterial genome. And what happens if it integrates? The recipient bacteria was having an allele A. On the other hand, the donor DNA has got A plus allele. And once these single-stranded A plus DNA gets entry inside the bacterial cell, there exists 3 strands and recombination by double crossover takes place between the donor DNA or the exogenode and the existing A allele. Then we will see that this leads to generation of a heteroduplex of A and A plus, and when the cell undergoes division, we get 50% of cells which are transformants wherein this A is being replaced by A plus and 50% of the cell remains non-transformed. So this is what is happening during natural transformation. There are actually 2 different methods of executing artificial transformation in bacteria. Number 1 method is physical method, which comprises of electroporation. It comprises of cuvette wherein it has 2 electrodes. In between these 2 electrodes, actually we have placed the mixture of competent cells and the DNA. An electric field is applied across the cuvette. Once electric pulse is given, this electric pulse allows the entry of the DNA inside the bacterial cell. This method is very good and it takes just 2 to 3 minutes to execute the experiment. Its efficiency is very high, but the problem lies here that it needs use of sophisticated instruments which are very expensive, and it is not possible for every lab to have a setup of electroporation machines. This method was first designed by Stanley Cohen in the year 1973, wherein he used calcium chloride and heat shock as the methods to make the cell competent and transfer of DNA into the bacterial cell. It mainly comprises of 2 steps. First is treating the cells with calcium chloride. This is to make the cell competent. Next, we are adding the plasmid DNA, which binds to the exterior of the cell, and we give it a heat shock of 42 degrees centigrade for just 2 minutes. This leads to the transfer of the plasmid DNA from the outside of the cell to the inside of the cell. So this is supposed to be the easiest method which is used mostly in the labs. So if we are carrying on transformation, we should be knowing the formula or method of calculating transformation efficiency. So basically, what is transformation efficiency? It is defined as the number of colony forming units or CFUs that are produced by 1 microgram of plasmid DNA. First of all, we need to find out the transformant CFU, which can be calculated by multiplying the number of colonies that we are getting in the antibiotic plates by the dilution factor, and we also need to multiply it with the original transformation volume divided by the plated volume. And the final transformation efficiency is calculated by dividing the transformant's CFU with the plasmid DNA.
Ma'am, what are the factors which regulate transformation efficiency?
The factors that are mostly regulating transformation efficiency is growth period of the bacteria. Means the bacterial cells that you are using for generating competence should be maintained in their log phase. If the cells are taken before the log phase or if they exceed the log phase, then we have seen that transformation efficiency decreases tremendously. The second factor that is playing important role in regulating transformation efficiency is temperature of competent cells during preparation. So, while preparing competent cells, it is said that strictly the temperature should be maintained less than 4 degrees centigrade. The third factor is the concentration of calcium chloride. Usually, the method of choice is to use 100 millimolar of calcium chloride is used to make the cells competent. And the fourth factor, that is the bacterial culture media. Bacterial culture media means after growing the cells, we make them competent and we are growing them in, say, Luria bertani broth, and if we grow them in a much more enriched media, say sock media, then we will see that the competence is acquired at a much higher rate. So, in all, we have understood the importance of these 4 factors in regulating the transformation efficiency.
So, ma'am, how do we isolate the transform cells?
Yeah, we do have got various methods of isolating the transformed cells. The first method is the selection of transformed cells. The plasmid that is received by the bacteria usually contains an antibiotic resistance cassette. So, the cells that gets transformed show resistance towards antibiotic and give colonies in antibiotic plates. On the other hand, the major population that remained untransformed are actually sensitive towards antibiotic, so they won't give any colonies in antibiotic plates. So, the plating is done after transformation in presence of antibiotic to screen out the transformed cells. On the other hand, there lies a problem that if we have introduced some DNA inside the plasmid, how we are going to screen out the cells that have received the plasmid plus insert and the cells that have received the only plasmid. Herein, we will take the example of PBR322, which has got 2 resistance genes for ampicillin and tetracycline, and the foreign DNA is introduced inside the tetracycline resistance cassette. So, when these 2 populations are getting transformed inside the bacterial cell, the cells that have received the only plasmid will show resistance towards ampicillin as well as tetracycline. On the other hand, the cells that have received gene of interest will show only resistance towards ampicillin as they become sensitive towards tetracycline because tetracycline resistance cassette got ruptured because of introduction of foreign DNA. So, finally, we will see that by method of replica plating, we can differentiate between the cells that have received only plasmid and the cells that have received plasmid plus insert. The requirements for the experiment are the chemicals that we need is Luria broth. Next, we need Luria agar plate containing ampicillin at a final concentration of 50 microgram per ml. Then we need calcium chloride solution, which should be autoclaved and at a concentration of 100 millimolar. We need autoclaved sterile water, and the bacterial strain that we are using is XL1 blue and the plasmid is PUC19. Next, the instruments and the glasswares that we need to use are the laminar airflow, as because the steps should be done under sterile conditions. Next, water bath incubator at 42 degrees centigrade for giving the heat shock. Then we need cold centrifuge to make the cells competent at 4 degrees centigrade. We need autoclaved sterile culture tubes for growing the cells. We need autoclaved sterile epigrafts, microtips, micropipette, and most important, a crushed ice cooler to maintain ice, wherein the entire step of competent cell making and transformation is to be carried out. We need to make 500 ml of lurea agar media, which is usually prepared by measuring desired amount of Lb, dissolving it in water, maintaining the pH at 7, and finally autoclaving it. Then this media is cooled down to a temperature of 40 to 45 degrees centigrade. After that, we will add ampicillin, because ampicillin will be destroyed at temperatures higher than 45 degrees centigrade. So we will pour the plates so that the final concentration in the plates is 50 microgram per ml. Next, the plates are poured and allowed to cool and solidify. The story starts with the Lb agar plate containing the starter culture of bacteria, wherefrom we will take a single colony and we will inoculate it into a small culture of 5 ml Lb broth. Then this is grown for overnight at 37 degree incubator shaker. Then the saturated culture that we are getting after overnight growth, therefrom we will inoculate say 500 microliter into a 50 ml Lb broth. These cells are grown for 2 to 3 hours at 37 degree incubator shaker until and unless the cell reaches the log phase, which is measured by measuring the OD of the cells in a spectrophotometer at OD is taken at 600 nanometers. It comes round to be 0.5 in case of E.Coli XL1 blue cells. Once we reach this OD, we need to lock the cells in this log phase. That's why we freeze it on ice, and we will see here that we have got the log phase cells. We have freeze them on ice for 10 minutes or more. After that, we centrifuge the polypropylene tubes at 6000 rpm for 10 minutes. After that, we get a cell pellet and the supernatant. We discard the supernatant and we save the pellet. These steps should be done precisely inside laminar airflow. Next, we add to it half the volume of calcium chloride. So if we have started the story with 1.5 ml of bacterial cell suspension, then the pellet that we will get will be suspended in 1.5 divided by 2, that is 750 microliters of 100 millimolar cold calcium chloride. We will make the suspension of the cells in calcium chloride, we will keep it on ice, we will again centrifuge it. Then we will discard the calcium chloride, and the cell pellet that we are getting is now to be suspended in say 100 microliters of calcium chloride. So this is how we have made the cells competent and they are just ready to uptake the plasmid DNA. The calcium chloride treated competent cells are mixed with ampicillin resistance cassette bearing plasmid PUC19, and the entire thing is incubated on ice for half an hour. This step actually allows the precipitation of DNA as calcium salts onto the surface of the cell. After that, we need to give it a heat shock at 42 degrees centigrade for 90 seconds to 1 minute. In this step, actually the plasmid DNA gets entry inside the host cell. After that, we need to immediately chill the cells on ice, keep it there for 5 minutes. After this step, we will be giving the cells a recovery as they have undergone tremendous amount of stress by remaining in calcium chloride, being given heat shock, and they have been introduced with a foreign DNA. This recovery is done usually in normal Lb broth devoid of antibiotic. After recovery, we will spread the cells onto the Petri dishes containing ampicillin as the resistant antibiotic. Ampicillin is present in the Petri dish along with LB. We have also shredded XGAL and IPTG. As I said, that PUC19 also bears fragmented LAG-Z plus gene, and this leads to formation of the blue colonies, and we will see the colonies by incubating the plate in an incubator for say 16 hours. The next day we will see blue colored colonies emerging on the plate. So this is the entire step of transformation. To estimate the dilution factor, to estimate the amount of competent cells that have been plated, and to take account of the total amount of cells that were made competent, as these values are going to help us in calculating the transformant CFU. And once we calculate the transformant CFU and divide it by the amount of plasmid DNA added, we get the value of transformation efficiency. We need to focus on the precautions that should be taken while performing transformation experiment. Number one is, while discarding the supernatant after centrifugation, the pellet at any cost should not be disturbed. Number two, the heat shock should not exceed from the allowed time limit. Number three, during centrifugation, the time and speed must be maintained properly, and while making cells competent, it should be strictly maintained at 4 degrees centigrade. Otherwise, there will be depletion in the cell competence, as we discussed in our lecture. And all the ingredients used in the process of transformation should be made perfectly sterile by autoclaving or some other means. In today's lecture, we have precisely focused on the 3 different means of introducing foreign DNA inside bacterial cells, and we have also discussed the importance of gene transfer in bacteria. We have discussed about the 2 different ways of artificial transformation. We have also discussed on the factors that regulate the transformation efficiency. We have precisely focused on the methods of selecting the transformants and the methods of selecting the recombinants. After that, we have gone through the elaborate steps of performing calcium chloride mediated transformation of E. coli XL1 blue cells with a plasmid called PUC19 containing a LAG-Z gene, fragmented LAG-Z, and ampicillin resistance cassette. Therein, after transformation, we have seen the emergence of blue colonies. We have counted those colonies, and we have gone through the steps of calculating transformation efficiency.